All living organisms on Earth needs energy to survive. This energy is present in the form of potential energy and is found in the bonds of a molecule called ATP. ATP is produced at a cellular level by the mitochondria and through a variety of ways, either through cellular respiration, fermentation, or ketosis. But in this video, we'll just be focusing on fermentation, specifically alcohol fermentation of the species Saccharomyces cerevisiae, also known as brewer's yeast. As I mentioned before, yeast produces its ATP through the process of alcohol fermentation, but let's break that process down into steps. First up, we have glycolysis, which is the first step for both cellular respiration and fermentation. We can see that glucose is converted into pyruvate, and in that process, NAD+ is reduced to NADH, as well as 2 molecules of ATPs are made. Now, if oxygen was present, then pyruvate will be converted into acetyl-CoA, which then will continue down the process of cellular respiration. If oxygen isn't present, then pyruvate will be converted into acetaldehyde through the process of decarboxylation. And a byproduct of this reaction is carbon dioxide. Acetaldehyde will then be converted into ethanol, and the NADH at the beginning will then be oxidized again into NAD+, and this NAD+ will go back to fuel glycolysis, and the cycle continues. So we can see that through the process of alcohol fermentation, yeast not only produces ATP, but also ethanol and carbon dioxide. Ethanol, a product of alcohol fermentation, is a biofuel, which means that it is derived from biological or organic materials such as plants or animals. Ethanol is used by many manufacturers and industries around the world for pharmaceutical purposes, for cosmetics, and of course, the beverage industry. Since ethanol is so widely used, finding the most efficient, low waste, as well as cost-effective method to produce it would be a game changer for those industries. Which is why my lab partners and I have set up an experiment which we tested the fermentation of yeast on different sources of carbohydrates. The carbohydrates will be parts of corn such as the kernel or the stem, or a derivative like corn syrup. The main difference between the carbohydrates, or we'll call them substrate, is that they all have different compositions of carbohydrates, which means that they all have varying amounts of glucose, sucrose, starch, or cellulose. We also added a water control group to make sure that the yeast does not react without the presence of a substrate. Carrying on with the experiment, we had 4 different durum vials containing the substrate and yeast. We took the measurement at the start, which was 4.4 for each of the durum vials, and every 5 minutes we would take measurements again. How this will work is that as the yeast react with the substrate, it will produce CO2 and ethanol in a 1-to-1 ratio. So if we measure the amount of CO2 that is produced and trapped in the vial, we can roughly equate that to the amount of ethanol produced as well. Let's move on to our result. We can see that with corn syrup, yeast produced the most amount of CO2. It also has the highest rate between 10 to 15 minutes, jumping from 0.6 to 3.5 milliliters. The reaction even finished 15 minutes prior to our set end time at 45 minutes. The vial with yeast and kernel had a steady production of carbon dioxide over time, ending at 2.6 milliliters. The production of CO2 for yeast and stem is also very steady, but it is significantly lower than corn kernel, ending at 0.4. And as expected, nothing changes with water. Here I have the line graph of the linear CO2 production rate of the yeast and kernel group. I did this by drawing a line of best fit through the production over time graph. Then I picked 2 points on the line and plugged their coordinates in the slope formula. I picked corn kernel because its CO2 production was the most steady, so it would be the most accurately represented with a linear graph. Now I did the math, and the linear rate of CO2 production for corn kernel is 0.06 milliliters per minute. Now let's go back to our result. Based on what we know about the varying compositions of carbohydrates in our substrate and the result, we can say that yeast prefers simple sugars over longer chained polymers during its process of fermentation. We can see that corn syrup has the highest rate of CO2 produced, even more than the other 3 combined, and that is because pure corn syrup is basically 100% glucose, a simple sugar. Corn kernels contain more sugar than stems, although they have very low amounts compared to corn syrup. Otherwise, they're mostly comprised of starch and cellulose. In conclusion, I think that biofuel is going to play a big role in our energy industry, finding a more efficient source of fuel. We can see in the experiment that all parts of corns, even the part that we don't eat, such as the stems or the cobs, can be used to make ethanol. And if we were able to do that on a global level, not with just corn, but with other organic materials, I think that it would help our environment a lot. And to wrap it up, I would like to thank my lab partners, Sarah and Brooke, and my TA, Melissa, for helping and guiding me through the experiment. Thank you for watching.